So before we get into topic 3, I want to talk a little bit about the library assignment that goes with lab 2. So there's a few things being asked in this library assignment, and one thing that's being asked is that you locate a journal article on a database called Pubmed. So Pubmed is it's a science searching database. It's mostly life sciences and biomedical kind of things. So if you're looking for chemistry, there might be some chemistry there, or physics. It's not as much focus as biology and medicine. But it's put together by the American government, and it is a very good database. What I'm going to do is click on this and show you how to find a paper. So I just clicked on that. I'm going to have to do a new share. There we go. So this should be loading up here. So this is Pubmed. This is what it looks like here. And what I'm going to do before I go on Pubmed is, let's see here. I'm just going to go back to, what is it, NC. There we go. There it is. So Pubmed is actually part of these greater databases here from the National Institutes of Health or the National Center for Biotechnology Information from the American government. But it's kind of international. These databases are all kind of linked through governments of different companies. There's all sorts of tools on here, genome databases, tools for analyzing DNA, all those kind of things. You can see Pubmed as part of it over here on the right. So here's Pubmed. There's basically a search bar, and you can do an advanced search if you were looking for certain authors and things like that, but you can kind of put whatever you want into the search bar. So I'll show you how that's going to work. What I want you to do is look ahead to lab 3 and see what it's about. And you'll see that lab 3 is about membranes. And we're going to be looking at the effects of different temperatures and solvents on membranes. So let's try membranes at temperature effects. So you can search for something like that. The more words you put in, the more specific it's going to be. So I'll hit search. And let's see what comes up. So if you take a look, 31,199 results. So lots and lots of papers here. They generally, sometimes they sorted out, last time I used the database, at least the last few times, it was sorted out by date. I'm not sure why they're not doing that, but you can see there are a whole bunch of papers here, more than 31,000. So what you probably want to do is, a lot of these will be only accessible if you have a subscription, right? So that means you have to have them in your library or you've subscribed to it. So what you can do over here on the left is you can select, it says free full text. So a lot of these are freely available online as well. So I selected the free ones, and that actually narrows it down to 10,000 results. So what you can do is click on one of these papers. So pick one that you think might be kind of like, has something to do with our lab. It doesn't have to be exact. I'm more or less looking that you can find a paper and that you can reference it properly. Whether it's exactly what we're looking for is not really the point. So let's click on this one here, hydrostatic pressure and temperature effects on the membranes of a seasonally migrating marine copepod. I don't know what a copepod is. Sounds like a fish or something. So you can click on this, and you can see there's a bunch of information about the paper. PLOS 1 is the journal title, gives you the date of publication, the journal volume, the DOA, that's the digital object identifier. So there's lots of information here. You can see the title of the paper, the name of the authors. The abstract is kind of a summary of the paper. And in this case, it's giving me some of the figures. And if I want to read the paper, usually, sometimes it's actually found directly in this link. Sometimes you have to click a little further, but often the full text links are found over here to the right. So it looks like there's 2 options. I can go to the journal database, and there's some other database here. So I'll click on this one here and we'll see what comes up. And it looks like there's the paper. So if you want to read the paper online, you can just scroll down and go through the whole paper like that. You don't actually have to read the paper for this class. Like I said, it's more or less a referencing and finding a journal article assignment. So what I want you to do, though, is find the PDF. So the PDF is usually another click away. You can see it's right over here on the right-hand side. And that's what I want you to do, is click on that, and you're going to download that. So I think in this case here, when I clicked on it, it downloaded here to my computer. You can see it's right there. And you're going to submit that for 1 mark of your assignment. And then what you're going to do is show me how to reference it. And I'm going to show you an example here. I'm just going to flip back to my PowerPoint and show you an example on how to reference it. So that's how you use PubMed. Should be pretty straightforward. Just follow the instructions in the lab manual and answer the questions and find that paper. So here's an example of another paper. This is one that I authored a few years ago, and this is what the PubMed citation looks like when it first comes up in the search. And just want to point out some of the features there. So there's 2 authors. You can see that's me and Anthony Clark. And there's the journal name. It's usually found somewhere. There's the year. I don't really care about the month, but there's the volume number. There's the page numbers. And you can see up there is the title of the paper. And the last thing is that digital object identifier. That's kind of a newer thing. You don't see that sometimes on older papers, but they're starting to add it now. And probably a lot of standard referencing formats are going to require it. Now you can include it or not include it. It doesn't really matter to me. When I click on the paper, I'll show you what the paper actually looks like. So that's the top of the paper there. And notice this now has a little bit more information. It has proper punctuation and things like that. So there's the journal name. The previous slide showed the abbreviation of the journal name. Again, the year, the page numbers. Sometimes this information is found at the bottom of the page, by the way. The volume numbers over there. Usually if you look for it, you can find it. The title of the paper, the authors again, the abstract, the digital object identifier. So notice I'm not pointing out where these people are from, right? This was at the University of Guelph when I was a graduate student. That's not really relevant to my referencing and citations. The author names and the journal title, these are important information. So I'll show you how you're going to cite and reference this now. So first, one of the questions asks you for a citation. So some people don't seem to know what a citation is. A citation is where you're referring to a work. And so I'll show you how it looks like, but usually it's found at the end of a sentence. There's different ways to do it, and I'm going to show you how you are going to do it in Biology 107. So what you do is you write these sentence information. So the information, my paper was about a bacterium called Pseudomonas aeruginosa and something called the penicillin binding protein. So maybe I would say something like penicillin binding proteins are involved in the cell wall construction in bacteria. And then that's the sentence. And at the end of the sentence, before the period, because it's still part of the sentence, you're going to have the names of the authors. Now, just the last names. So last name 1, last name 2, followed by the year of publication. So in this case here, that's what it's going to look like with that particular paper. There's 2 authors, and the year was 2008, and the period is found at the end of the brackets. Take note, not all your papers are going to look like this. Some are going to have 1 author, some are going to have 5. Take a look at Appendix A in the lab manual, and there are more examples there on how to do this with a little bit more of a description. Everything I'm telling you, by the way, about lab reports is all in the lab manual somewhere. What I'm trying to do is just dole it out in little bits and pieces. So if you have, just want to show you what it's going to look like if you have different numbers of authors, right? So generally 1 author is exact same format, and it's just last name followed by the year. So that should be pretty straightforward. 2 authors I just showed you is here have the word "and" between their last names. And then 3 authors, we're actually using, we're not going to write them all out. Actually, some journal papers have 20 authors. So you're not going to write all 20 names out. So usually the idea is when you have 3 or more, you're going to use et al. So et al is Latin, and it actually means et alia, or that's an abbreviation for et alia, which means "and others." So since it's Latin, we're going to make sure it's in italics, and that's the format we're using here. You don't always see et al in italics, but this is the format we're using in Biology 107. So it might look something like this: Jones et al. Notice there's punctuation there. We have a period, right? The punctuation is very important when you're doing referencing, and then followed by the year, and the period goes at the end of the sentence. So that's the citation. That's one of the questions. It's asking you to write a sentence. I don't really care what your sentence is. I'm just looking to see that you can do the citation properly. And then the last part of referencing is giving me the full bibliographical information in a reference list. So this reference list, we actually call it a literature cited or a works cited section, meaning this is the information, this is the works, the journal articles or whatnot that I have cited. And so journal articles look like this. This is the format. So you have all the last names of all the authors, followed by their initials. Notice there's punctuation in there, commas and periods, followed by the year, followed by the title of the journal article, journal name, volume number, and pages. And sometimes, like I said, people are starting to add that digital object identifier at the end, which is not necessary, but you can include it. So for the example that I showed you, it's going to look something like this. You can see the two authors. Like I said, take a look at all this punctuation here. You don't include the punctuation, it's wrong, and you're going to lose marks. Notice the title is in sentence form, so it starts with a capital, and then everything else is small letters, with the exception of Pseudomonas aeruginosa. That's a species name, and so species name has to be spelled properly and in italics. The journal name can be the abbreviation, or you can use the full journal name, Journal of Bacteriology, in this particular example. Notice the volume numbers in bold, and the page numbers are not. So just keep in mind, as I said, punctuation and details are very important, because if you don't do it, you don't have it correct. There are other citation methods. There's Chicago style, APA, MLA. There's probably 100 different ways to do it. So make sure you do it the Biology 107 way, and you'll get full marks. Should be easy marks. And then you'll do that again on your formal report for Lab 3 as well. So that is part of that literature assignment that you're going to do with part of Lab 2, Lab 3 pre-lab assignment kind of thing. There's a couple other questions in there. You should be able to find the answers in the lab manual, and I'm not going to spend any more time on that. I'll talk a little bit more about lab reports next on Monday as well. I'll talk a little bit more about some of the specific questions. Okay, so let's talk about topic 3. Topic 3, we're going to talk about microscopy today, and that will take most of the lecture. We'll get to some of these other things. We'll spill over until next week. So we've had a little bit of an introduction to microscopy in the lab, and I showed you a picture, something like this, similar to this at the beginning, maybe in our first lecture. And what this is showing, you can see there's all these things here, and we can see quite a few things with our human eye, but there's so many things that we cannot see with our eyeballs. And a lot of these things are in the realm of cell biology. So we're talking about most cells, animal, plant, or bacterial. We need some sort of microscope to see them. If you get down to smaller things like viruses, you can see here's a virus right there. In fact, we're going to need an electron microscope for that. One more thing that we will talk about as well that's not mentioned in the textbook yet is something called scanning probe microscopy, which is a pretty cool kind of newish technique, and we'll get to there. If we don't make it today, we'll get there on Monday for sure. So let's talk about a microscope. What do we need for a microscope? So two of the things are right there. Number one, we need a specimen. So your specimen, in the case of a traditional microscope, is something that you put on a slide. Number two, you need some sort of a lens. Maybe number two actually should be what I'm writing down as number three. Number three is an illumination source. That should have been number two. So that is a basic microscope. So if you take a look, there's my object and my lens. The light is going to go through my object, through the lens. And what it does is presents your eyeball with a larger image. So notice I didn't say light source. Not all microscopes use light. Some microscopes use electrons. So that's why I'm kind of using something that is more of a catch-all. And we'll talk a little bit about some of the details of the different types of microscopes for sure. So here's a little bit of a kind of a quick history of microscopes. You can see on the far left is the Van Leeuwenhoek microscope. As I mentioned, it's not much more than really a sophisticated magnifying glass. The lens is found right there. This contraption is just holding the specimen in place and then adjusting it so you can focus it. He was holding it up to the light. the sunlight, right? In the middle, we have a compound microscope. So compound microscope means there's two lenses. So you've got an objective lens down there, an ocular lens up there. So two lenses can help magnify things a little bit better. This microscope was used by Robert Hooke, and this is basically an oil lamp down there, which is providing the light, but not necessary. If it's bright enough, if it's bright enough in the room, you don't need an illumination source. Most modern light microscopes look something like this, and you can see they have multiple objective lenses, so you can zoom in or zoom out on your specimen. And that one that's shown there is a pretty basic kind of cheap looking one. You know, don't be deceived. Sometimes microscopes may look cheap, but they can be quite expensive depending on what materials are made out of. This is kind of a zoomed in view of kind of looking at what's going on inside one of these microscopes. Most modern microscopes are a little bit more ergonomic. If you take a look back at Hooke's microscope in the middle, it's not very comfortable to look at. Whereas a lot of these microscopes, the light doesn't go straight up, it goes and gets bent through a prism. Sometimes there's two eyepieces to kind of help it make it easier on the eye, and those kind of things. So a lot of bells and whistles. Most microscopes now have some sort of light source at the bottom. They have these condenser lenses that focus the light, and all those things help with the design of it to hopefully give you a better image. So I guess we already looked at this video. I'm just going to keep moving on, and there's our microscope there. And just to point out a couple of things, most people have probably found these. This is a diaphragm there at the bottom that can control how much light goes through the specimen. The stage adjust knobs are right there. They're not really shown very well in that picture. And of course, these are the ocular lenses, and these are the objective lenses. So I think most people have gotten reasonably familiar with those, and we'll get a chance to use them a couple more times this semester. So one thing that we are going to get a chance to do in Lab 4 is we're going to get a chance to do a procedure called oil immersion. So with this microscope here, some of you may have played around with 100 times lens, which I'm not sure if that's the one here, the big one anyway, with the white band on it. When you're starting to look at really small samples, what happens, I'm just looking at this red line here. Okay, the light, the light goes and it goes through the glass slide, and every time the light goes through a different medium, it can be refracted a little bit. So anytime you're looking at something underwater, for example, sometimes it just doesn't look right because the lines of light aren't coming straight at you. And that's kind of the same idea here. So a lot of light actually scatters, and the specimen looks relatively dark. So what we can do is we can, with this lens, we can add oil. So this oil is literally a special type of oil that's used to put on the specimen. It makes contact with the specimen and the lens. So we're looking at this blue line here now. And that allows more light to pass through the specimen. So you get much more light and a sharper image. And it's really essential for using oil immersion when we're looking at bacteria. So we are going to look at a video talking about oil immersion. I'm not going to do that today. What I'm going to do is actually play that video for you at the beginning of Lab 4, so you have an idea how oil immersion works. So what I want to do now is talk a little bit about what we can do to make a better image, right? Everyone's had this kind of experience with cell phone cameras or traditional cameras, and sometimes you get a good image and sometimes you get a bad image, and there's a lot of ththings you can blame. Sometimes it's the person taking the photo. Some people just aren't good at it. It's kind of a fact. Sometimes it's the phone or the camera. Some of newer ones tend to be better than older ones. So I want to talk about what's going on and take some of those principles and bring it to microscopy. So the first way we can improve an image is magnification. So on our microscope, we have 4 objective lenses, right? So they're down here shown in the picture, and the bigger ones usually mean you're zooming in more. So same idea with a camera. If you ever see somebody with a professional camera, that guy with a really big lens, it's a big zoom lens, right? And so he or she is going to be able to get in closer to the whatever you're trying to take a picture of. and you're gonna get a better image, at least a higher magnified image. That's one thing we can do to improve the quality of an image, is just change the lens. And sometimes even having more lenses, 2 lenses versus 1, compound versus a regular microscope. Second variable, oh, there's the objective lenses, by the way, from our microscope. So you can see we've got 100 times, 40 times, 10 times, and 4 times. And they all have their uses depending on what you're looking at. If you're looking at an insect or a bug, it might actually be better to use the 4 times so you can see the whole specimen. If you're looking at a bacterium, you're going to want to zoom in and use the 100 times for sure. Number 2 is something called resolution. So resolution is specifically, it's a number, and it's a number talking about the minimum distance between 2 distinguishable points. Another way to think about resolution is clarity. So you can imagine, you know, some people with cell phone cameras, they like to zoom in and then they look and they're just like, what is wrong with this photo? And that's because it's not clear. You can zoom in all you want. It doesn't necessarily mean you're going to get a good image. So I'll show you an image here, or 2 images. And you can see on the left, we have one microscope, on the right, we have another microscope. It's the same picture, but clearly there's a very big difference. Microscope B is sharper. I can make out each of the cells. I can distinguish each cell as distinct individual cells. Whereas on the left, even though I have the same magnification, I can't necessarily distinguish the individual cells. So you can see on the right, we're talking about the resolution being 0.2 micrometers. So a smaller number means we can see smaller objects with more clarity, is really what we're talking about. Whereas the one on the right, 2 micrometers, which is not enough to make out those individual bacteria. I have a little cartoon for you. You can see this is the far side, and they're looking at a photo show, and he says, no, wait, that's not Uncle Floyd. Who's that? I think it's just an air bubble. So resolution is clarity. And this is where, again, going back to the cell phone analogy, newer cell phones have better lenses, which usually lead to better clarity. There's also some software things you can do too, particularly with cell phones and a lot of modern technology that kind of help with that kind of thing, you know, in terms of stability and whatnot. But resolution is basically clarity. So how do we get better resolution? That is the question. You know, I'd mentioned that newer cell phones tend to be better than old cell phones. What about microscopes? We've had microscopes for hundreds of years, and we've got some pretty good images from hundreds of years ago. So what is the difference? So here's where I want you to think about a different kind of thing. Is imagine I bought a microscope from Walmart, and it's a $50 toy microscope. And then I have a microscope from the Chiana lab, which cost $8,000. Which one do you think is going to be better? Probably everyone is going to choose the Chiana microscope. So what's the difference between these things? Well, think about the toy microscope. What is it made of? It's made out of plastic. So that's actually a big thing, is that plastic is not the best lens material. Turns out that glass is better than plastic. And it turns out that quartz, so quartz is a mineral, quartz is better than glass. So in the Chiana microscopes, we actually have quartz, and each of those lenses are worth, I think they're hundreds of dollars each. And that's what's making a big difference. A bigger part of the technology is the type of lens material. Another thing we can do is we can use smaller wavelengths, and that's where we're going to get into electron microscopy. And so we'll get to there in a couple of minutes. You can see number 3, the last thing we can do to improve our images is do something called improve the contrast. So contrast is the difference between the parts of a sample, right? So I'll show you a picture here. You take a look. We've got 2 shots of probably the same cell, or at least the same sample. The one on the left is not stained. You can still make it out. It's pretty good. It's sharp. You can see the nucleus, but the one on the right is so much better. A little bit of stain helps with contrast, and we can make out the parts. Some things are almost invisible without being stained. So staining improves contrast. That's kind of the big message here. And you probably saw that from the previous slide. Here's another picture. You can see these are some organisms called Giardia. I guess I can write that down. This is called Giardia lamblia. And this is an intestinal parasite, and it's kind of cool to look at, right? It's got 2 nuclei, and they sort of look like little aliens, but very, very nice stain. So one type of stain we are going to do in lab 4 is something called a Gram stain. And the Gram stain was named after this guy, Hans Christian Gram, and he was looking at bacteria. And this is a really important stain, both historically and still used today in modern science and medicine quite regularly. So it's a very, very important stain. What it does is distinguish 2 different types of bacteria, and we call them gram negative and gram positive. Gram positive are purple after the staining procedure, and gram negative are pink. So we'll come back to why that is shortly when we get into cell walls, and when we talk about bacteria. We'll talk about exactly why they stain different colors when we get there. But you're going to make a Gram stain. It's going to look something like that. Okay, so moving right along, I wanted to talk about kind of one more type of light microscopy, something a little bit more advanced, something a little bit more expensive. So I mentioned that the microscopes in the Canada lab are maybe $8,000 each or something like that. Probably in today's dollars, they're maybe $10,000 or $12,000. You know, that's expensive, but it's affordable enough that a college can buy, you know, a handful of them and whatnot. One way to improve our sample is to use fluorescence. So fluorescence is just using a fluorescent stain. And you probably know a little bit about fluorescence, is that fluorescent things glow. So now, rather than just having a specimen that's one color, another color, it's actually glowing. And you can get some amazing images, and you can get some really sharp details from this kind of thing. Other than that, it's kind of similar. There's different types of fluorescence microscopy. You can see this one here is using a laser, and the costs, you know, they range from maybe $25,000 to $100,000 or $200,000, depending on what you're buying. These confocal ones, for example, they use lasers, and they have some special techniques. I'll talk about them in a minute, and expensive software. So they tend to be like $100,000 or something like that. So now you're starting to get into, you know, some research grade equipment that's going to cost you a little bit of money. You're going to need a research grant for that kind of thing. Let me show you some pictures of some fluorescence microscopy. So there's some Staphylococcus. Remember, Staphylococcus is clustered spheres, and you can see that they show up very nicely in that image. Check out some of these other images here. They're just absolutely incredible. There's some epithelial cells. You can see the nuclei are stained in one color, and the cytoskeleton is stained in another color. And you can make out the cells. They're just it's just stunning, and the detail is really quite incredible. There's our Giardia again, still looking like a little bit of an alien. And there's some spindle fibers and chromosomes, and they're at metaphase, and they're getting ready to separate and do their thing during mitosis. I'll show you more pictures. I have lots of images to show you. Here's a blood smear, and in this case, what they're doing is looking for this organism, Plasmodium. This is the organism that causes malaria, and so they can be used. This type of technique can be used for diagnostic purposes, and it's not unheard of nowadays for a hospital to have at least one fluorescent microscope nowadays for this reason, because it is a very simple technique to use, and there's no question right there of whether malaria is found in this particular blood sample. Just some other interesting ones I found on the internet. I don't know what they're doing here, but they made a rainbow cell. I thought that one was kind of pretty. You can see all those individual fluorescent proteins on the surface of the cell. You know, decent amount of detail and quite sharp. So I just want to talk about confocal microscopy for a moment. A lot of fluorescence now, for at least research, is moving towards this confocal microscopy. That's using the lasers. So why are we doing that? It looks like we're getting a better image. So one of the problems with specimens is that they're in 3 dimensions. And if you have a particularly large specimen like a eukaryotic cell, Sometimes, you focus in on the nucleus and it makes this other part of the cell go blurry. You focus on the other part of the cell and the nucleus goes blurry. And so that's kind of a problem. So you end up with a blurry image overall. So the confocal microscopy, it uses a laser and it shines in and just focuses in on the parts that you need. And then often what you can do is you can actually compose those parts together using software and get a much, much sharper image. So a lot of the really amazing pictures nowadays are done with confocal microscopy. And I'll show you a few more of those pictures as well. Some of them are just incredible. This is a contest done by Nikon. They make cameras, and just some stunning pictures here in this. This was a skin cell here, and I mean, look at that. It's just incredible. You can see all that detail, and some very interesting things to see. Here's the head of a tapeworm, and this is I think this is the beef tapeworm, kind of creepy looking. Here's some axons in an embryonic chick. They made them a whole bunch of different colors. I don't really know how they did this particular technique, but very, very stunning image. Like I said, some very cool things. Like I said, I just want to point out you can do digital enhancement nowadays with computers. And in this case, I think what they did is they took something like 100 photos and then used some sort of software to put it through and get a better image of that particular cell. So a lot of those type of things are done nowadays as well. The confocal microscopy can get a 3-dimensional image of something. Here's some, this is a yeast cell budding, and all those green things are mitochondria, and they mapped it out in 3 dimensions. So pretty cool stuff. A couple more pictures, holiday lights. This is in the brain in a zebrafish, and one of my favorites here, up close and personal. You can see the nucleus. All those yellow things are mitochondria, and I thought it was pretty cool. Okay, so we have magnification. We can improve that by having a bigger lens. Resolution, we can improve that by having a better lens material such as quartz. In contrast, we can improve that by staining. And the problem is there's a limit. There's a limit to how much we can do with visible light. So we're going to segue and start talking about electron microscopy in a minute, but what I want to do now is kind of stop and make a few notes. So what I'm going to do, usually I'm doing this on the board as I'm giving the lecture, but what I will do is exactly what I did before is I'm going to do this in a Word document. So let me just share that for you. So here's my Word document. So you can see what I have here is I'm going to fill out some of the information for light microscopy. And then later we'll come back and fill in some of the information for electron microscopy. So you can see we've got illumination source, lenses, viewing, color techniques, cost, ease, magnification, resolution, things like that. So let's fill in some of these details here. So light microscopy. Usually we're using visible light. Sometimes in fluorescent microscopes we can use UV light, and the light source can include things like sunlight, lamps, candles, whatever you want, lasers in the case of confocal microscopy. So maybe I will make that example. There we go. All right, so hopefully that's straightforward. The lenses, as I mentioned, they could be plastic, glass, or quartz are the most common materials. And quartz, of course, is the best. And there are other materials, lots of them nowadays, but quartz is kind of the best one to use. Viewing. So you can view with your own eyes, or nowadays, of course, we can use all sorts of digital cameras. So that's an easy one too. Color. We'll just say yes, all of your cells are going to be in color, and sometimes the colors may depend on your staining. Colors may depend on the stain used. Okay. Techniques. So what are some of the techniques that we talked about? We talked about oil immersion. There are other techniques. We have not talked about everything there is to talk about microscopy. This is just kind of an introduction to some of the techniques. Oil immersions, we talked about fluorescence. I think I just spelled that wrong, didn't I? At least the nice thing about this is there is the spell check. And we talked about confocal microscopy. So there's some techniques there for you. Cost and ease, we'll just say affordable. And easy to train someone, or easy to use, we'll say. Meaning that you know, we can plop a bunch of students into a lab and we can get them going with the microscopes in 5 minutes, kind of thing. Relatively easy to use. Okay, so magnification is about 1,000 times, maybe 2,000 times for advanced fluorescent techniques. And there's probably, we can probably push that a little bit more, closer to 2,500 nowadays with some of the advanced softwares and things like that we have. But for the most part, that is, that's about the limit. And the resolution, I just got to look that one up here in my notes. Make sure I don't get that number wrong. So just give me a second here. Okay, there it is. Yeah, that's what I thought. So resolution is approximately 0.5 micrometers. Maybe I will make that an actual micro for you. Insert symbol, and there it is. Okay, it's approximately 0.5 micrometers, which is 500 nanometers. If you think metric like I do, those numbers make sense. So just to give you an idea of how small that is, your average bacterial cell is about a micrometer. So it means we can see bacteria, but we can't really see any details. You know, maybe with that advanced fluorescent stuff, we can start to get down to maybe 0.1 micrometer or something like that. I'm not sure what the limits are that people are pushing nowadays. All right, so there is the first part of these notes. So hopefully everyone got that. If not, I'm going to come back to that in a few minutes once we talk about the electron microscopy. So let's talk about electron microscopy. All right, so like I said, you know, there's a limit to light in terms of the resolution and how small we can see things. If we want to see details around organelles and whatnot, we need to do something better. So what is that thing better is, well, there are limits around, visible light in terms of the resolution. So why don't we use smaller wavelengths? Well, smaller wavelengths include things like X-rays and electron waves. And so that's the whole concept behind the electron microscope, is using smaller wavelengths so you can bounce off smaller objects and see them more clearly. So if you take a look, here's kind of a schematic. You can see on the left is a light microscope. You've got the light source here. You've got a condenser lens. You've got a specimen objective lens, and there's your eyeball on the end. And you can see that image of the little mosquito head. The electron microscope. So what is different here? We're not using light anymore. We're using an electron beam. The lenses here. So if you take a look at these lenses, we've got a couple of lenses here. The lenses are actually magnetic fields. And that's kind of the big difference, right? And the last thing is you don't have an eyeball down at the end there. You're not going to stick your eyeballs in a high energy electron beam, but you're going to get still an image of the specimen, and the image is going to be on a photographic paper, or it's going to be a digital image put into a computer nowadays, is usually what we're looking at. And you can see the mosquito picture there on the right. So I want to just show you, talk about a couple of types of electron microscopy, because there's two main types that you should know. The first type is called scanning electron microscopy, and that's actually what is alluded to in this image here on the previous page. This is one that I used back at the University of Guelph. This is the actual microscope here, by the way. Everything else is basically a giant computer from the 1980s, believe it or not. The control panels are right here. over here on the right-hand side, it's basically being run off a typical desktop computer. But at the time, I think it was $100,000 in the 1980s, and it's working just fine. So why upgrade it? You know, nowadays you're looking at double or triple that price for something equivalent. So if it's working, there's nothing wrong with that. Here are some of my pictures. Notice they are all one color. There's no color of electrons. There are not green electrons. There are not blue electrons. So the colors come out in various shades of dark or lightness. So grayscale, we call that. You can add color later. That's what I did in the final picture on the bottom right. But the colors are all fake, just added afterwards, basically, to make it look nice. So it's all grayscale. The other thing you may notice from these images is that the images are, you're just looking at the surface of these things. So that's what a scanning electron microscope does, is it bounces the electrons off the surface and then they're detected. And that's how you get your image. And so you're only seeing the outside of these objects. I'll show you some more images here. Here's some more parasites, a hookworm. That one is absolutely terrifying. Every time I see that picture, I just think I would never want to be found in the dark with one. There's a tick, there's a tapeworm, another type of tapeworm. And you know, this is something that you see a lot, people looking at insects and bugs, because they're very, very interesting details. Kind of wish I'd thrown a mosquito on when I was doing the microscopy. Never thought about it at the time. So notice there's a note here. It says the specimen is coated with a heavy metal. So this is important because heavy metals have lots of electrons. So usually we're using gold or platinum or something like that. And this is a way of staining it. With lots of electrons, you can see the specimen so much better. Here's some more images of the electron microscopy. These are some of the early images of the SARS-CoV-2 virus being shed from cells. You can see the image on the left is the grayscale. The image on the right is in color. And so they've added the color to emphasize the detail. And it's just stunning. It pops right out at you. You can see all those little virus particles, those little nasty things ready to go. Here's another colorized one. This is showing the HIV virus budding from a white blood cell. And like I said, lots of nice pictures out there that people have done. Here's a really stunning one of E. coli, and you can see colorized. You can see all sorts of cellular features. So we're seeing a lot more features of these things than you would see with a light microscope. Here's a little bit of art, salt and pepper. So salt crystal and a peppercorn side by side. I thought that one was pretty cool. I'm just going to show you how the process works, and then maybe I'll take a couple of notes on that Word document here. And but let's take a look how this works. So there's your sample. This is actually a mosquito. This is not one that I did. I found this on the internet. And the first thing you do is you stick it in this instrument here. I have no idea how this instrument works, but it's called a sputter coater. And you press the start button. It takes about a minute, and somehow it spits gold particles onto your specimen. Usually we use gold. It's a lot cheaper than platinum. There's a very, very thin layer of gold on the surface of the specimen. There's the specimen there. You can see it's kind of looking a little shiny. It's just on this little platform, and then it goes in the microscope. So notice there's a big door here with some big handles, and the specimen is going to be put under vacuum. Now remember that air has electrons in it. So we want to pump the air out, and that way we're going to not have any interference, and we're hopefully going to get a much sharper image than what you would have if you didn't pump the air out. And then here's some images. You can see there's the insect's head. You can make out all those individual hairs, and you can zoom in, and so you can see there's some hairs. We're zooming in on some hairs there. Zoom in a little bit more, and we're making out some details. And these little things here are actual proteins on the hair. So it's just incredible. Look at those proteins, the detail we can get compared to something like a light microscope. Just absolutely remarkable. Here's a few more pictures. And like I said, I'll write down some notes for you in a moment. There's Giardia again. And there's some human blood that's been colorized and really pops out at you. There's some human chromosomes. Again, lots and lots of detail, which is really what we want. We love detail. One thing that is important to note about electron microscopes is we can zoom in enough to see actual viruses. And this is one way we can research viruses. Otherwise, we can't see them with a normal microscope. So there's Ebola again, and there's a bacteriophage, something that infects E. coli. And so these are things that are not visible under our regular microscope. All right, so there is another type of electron microscopy I want to talk about, but what I want to do is make a few notes here first, because I think we're running out of time, and then we'll see if we have time to talk about that other type of electron microscopy. So let's fill in some details here. Illumination source is an electron beam. Electron, I got to spell electron correctly. The lenses are magnetic fields. And the viewing is usually, nowadays, we'll call it, what would be the term I'm looking for? We'll call it a digital detector or digital image. But old school, we would use photographic paper. But most microscopes now have been converted to use some sort of digital camera or something like that. Color, no. These are in grayscale. We can add color later if we want as an option. So the techniques we're going to talk about are SEM, which is scanning electron microscopy, and TEM, which is transmission. So we haven't talked about the TEM yet, but remember the SEM, you're getting those three-dimensional surface images, and that's kind of one of the things that distinguishes it from the TEM. So cost, this is expensive. And it takes training. Took them, probably spent about 2 days of training just to use that scanning electron microscope. So we'll say lots of training. And if you're any good at it, then you are going to, you might even get paid a decent amount. So what is the magnification? Magnification for SEM is up to about 200,000 times. So that's amazing. TEM is up to 1,000,000 times. So we're really zooming in on things. And I think there's even some more newer advanced techniques that can even improve on that and double those numbers. I'm not up on that, but I'd have to check that. And the resolution, again, I just got to double check my number. I have a number in my head. I don't want to get it incorrect. So the resolution is very, very small. So the resolution is about 0.001 micrometers. So we'll insert. I can't get that symbol. I'm going to have to. Top of my screen is messing up here for me. So that's about 1 nanometer. So now we can see things as small as actual membranes. We can actually zoom in and we can see the thickness of a membrane. All right, just looking at my clock here, and it looks like I'm basically out of time. So what we will do is come back next day. We're going to talk about TEM to sort of finish off this little unit on microscopy. We're going to talk about probe microscopy and a few other things to do with cells and cell culture and things like that. Hopefully you enjoyed these images, and this is some very interesting stuff, and it's nice that I was able to see everybody yesterday in the lab, and hopefully you enjoyed that, and hopefully you'll enjoy your weekend.